Hello everyone, welcome to the series Bacteriology, and this is the first video in the series, and today we are going to discuss about classification of bacteria. Before discussing the classification, you must have the thorough knowledge about the basic structure of bacteria. So I will just start with a simple and a quick revision of bacteria, its structure, then move to the classification. Basically, bacteriology is the study of the bacteria and its related diseases. Specifically, in this series Bacteriology, we are going to discuss about the clinical bacteria and will discuss each bacteria individually with its characteristics, its clinical features, its pathogenesis and different diseases caused by them, what is their mechanism and how can we diagnose them, what are the different lab diagnostic criteria and specific culture media for each bacteria, their treatment and prevention and all that in the end. So before discussing the individual bacteria, it was must to discuss the classification of the bacteria, because then it would be easy to study individual bacteria once we know in which category it fits. So let's start. Structure of the bacteria. So bacteria, they are single cell microorganisms. They are among the smallest and simplest living organisms on the earth. Bacteria are classified as prokaryotes, which means they lack a well-defined nucleus and membrane-bound organelles. Their genetic material is contained in a region of cell called nucleoid. Structure of bacteria. In the structure of bacteria, bacteria have three shapes. These three shapes are cocci, bacilli and spirochetes. Cocci are the bacteria that have spherical shape, and bacilli are the bacteria that have rod shape, and spirochetes are the bacteria that shape is spiral. So this is the diagram which shows the different structures of the different shapes of the bacteria and how they are arranged. For example, if one cocci is there, so we would call it monococcus bacteria, and if two cocci are there, then we'll call it diplococcus, and if the cocci are arranged in chains, then it is known as streptococcus, and many more forms are there. So there are basically three shapes. Bacterial cell wall. In the structure of bacteria, we have bacterial cell wall, then we have bacterial cell membrane, and then cytoplasm. Inside that was the nucleoid. So in the classification, the most important is the bacterial cell wall. So bacterial cell wall, it is primarily made up of peptidoglycan, which is a complex structure of sugars and peptides. Cell wall provides shape and integrity to bacteria. It prevents the bacterial cells from bursting due to osmotic changes. Types of the bacteria on the basis of cell wall. From here we are starting the classification of the bacteria. So first, on the basis of the cell wall composition, bacteria are of two types, gram positive and gram negative. Gram positive and gram negative, this classification is based on the gram staining of the bacteria. Gram positive bacteria, they stain purple, and gram negative bacteria, they stain pink. This difference is due to the ability of the gram positive bacteria to retain the crystal violet iodine complex in the presence of lipid solvent, usually alcohol, in the gram staining procedure. And gram negative bacteria, because they have an outer lipid containing membrane and thin peptidoglycan, so it lose the purple dye when reacted with the alcohol, so it become colorless, and then they stain pink when exposed to the counter staining in the gram staining procedure. In summary, gram positive bacteria are those which stain purple or violet, and gram negative bacteria are those which stain pink. This difference is on the basis of the cell wall composition. Classification of medically important bacteria. Basically, the classification is based on the shape and the cell wall. So we discussed there are two there were three shapes of the bacteria, cocci, bacilli and spirochetes. So the main classification is on the basis of cocci and bacilli. And cell wall, whether the bacteria has a rigid thick cell wall, or it has flexible thin cell wall, or the bacteria has no cell wall at all. So there is one bacteria which doesn't have a cell wall, otherwise all the bacteria have cell wall in its structure. Rigid thick cell wall bacteria. Based on this composition, bacteria are of two types. This means that bacteria has cell wall, but it is rigid and thick walled. So they are classified as free living and non-free living. Free living bacteria are also known as extracellular bacteria, and non-free living are also known as obligate intracellular parasites. The examples include free living bacteria. Based on this composition, they are either gram-positive or gram-negative or acid-fast bacteria. So, what are gram-positive bacteria? Gram-positive bacteria are those which stain purple with the gram staining. So, they are either cocci or bacilli. In cocci, the examples are streptococcus and staphylococcus. In bacilli, examples are spore forming or non-spore forming bacilli. In spore forming, again we have aerobic or anaerobic. In aerobic, we have bacillus, and in anaerobic, we have clostridium. In non-spore forming, either they are filamentous or they are non-filamentous. Filamentous includes actinomycetes and nocardia, and non-filamentous bacilli includes corynebacterium and listeria. Gram-negative bacteria, they are also cocci or bacilli. In cocci, we have Neisseria or Moraxilla. In bacilli, we have facultative, aerobic and anaerobic bacteria. In facultative, we have either straight-shaped bacteria or curved-shaped bacteria. In straight, again we have respiratory, zoonotic or enteric bacteria. The gram-negative bacilli, the examples, facultative bacteria, they include straight or curved. In straight, we have either respiratory, zoonotic or enteric. In respiratory, the example of Haemophilus, Bordetella, Legionella, and in zoonotic, we have Brucella, Francisella, and in enteric, we have a list of bacteria. So, they are Enterobacter, Serratia, Salmonella, and Klebsiella, and Shigella, and Proteus, and E. coli. In curved bacteria, we have Campylobacter, Helicobacter, and Vibrio. In aerobic classification, we have Pseudomonas, strictly aerobic bacteria, and anaerobic, which are strictly anaerobic, they are bacteroids. Facultative means that the bacteria doesn't necessarily require the oxygen. They can survive in the presence of oxygen or in the absence of oxygen. Aerobic are the bacteria which strictly requires the presence of oxygen for their growth, and anaerobic bacteria are those which doesn't require oxygen. They can survive only in the anaerobic environment without the oxygen. Acid-fast bacteria, they don't take up the conventional gram staining. The example is Mycobacterium tuberculosis, and Mycobacterium leprae. Another bacteria, Nocardia, it is also acid fast, but it is weakly acid fast bacteria. So these examples are under the acid fast bacteria. Non-free living bacteria, they are also known as obligate intracellular parasites. They include Rickettsia and Chlamydia. Then the next classification, flexible thin-walled bacteria, they are spirochetes. The examples include Treponema, Borrelia and Leptospira. Wall-less bacteria, Mycoplasma. This is the only bacteria which does not have wall in its cell wall in its structure. Only cell membrane is present. This is the whole summary of the classification of the bacteria, and this is taken from the review of medical microbiology and immunology by the Warren Levinson book. This is the algorithm of gram-positive bacteria, and this is the algorithm of gram-negative bacteria taken from the book USMLE First Aid. I hope you understand the video. If you have any questions, feel free to comment down in the comment box, and thanks for watching.